This is my Unit 4 case connection by me, Jason Adrians, and today is 8/25/2024. So I'm going to be talking about case 1. This is the first case of the 4 that I'll be addressing throughout this video. Case 1 is about a newborn patient that has appeared normal at the time of birth, but then within 24 hours, ammonia levels in the blood were extremely elevated, leading to hyperammonemia, and then was back in the ER. Ammonia is highly toxic to the body, particularly to the central nervous system. Given its toxicity, even at slightly elevated levels, the body has developed metabolic mechanisms to remove ammonia, ultimately converting it into urea for excretion. One of the mechanisms that helps remove ammonia from the body is glutamate dehydrogenase, or GDH for short. GDH is an enzyme found in the mitochondria of cells, primarily the liver, but also in the brain and kidneys. GDH is a reversible enzyme that can take the ionic form of ammonium, combine it with alpha-ketoglutarate to form glutamate. Strictly speaking about GDH, this is how ammonia/ammonium is removed from the blood. Another way glutamate is formed is by a process called transamination. Transamination uses different enzymes called transaminases to combine an amino acid with alpha-ketoglutarate, a keto acid. In this case, the amine group on the protein is ammonia, which is transferred to alpha-ketoglutarate to form glutamate. Regardless of how the glutamate is formed, glutamate is then transported via the blood to the liver and ultimately the mitochondria of liver cells, where GDH converts glutamate back into ammonium and alpha-ketoglutarate. Staying in the mitochondria, ammonium combines with bicarbonate and ATP to form carbamoyl phosphate via an enzyme called carbamoyl phosphate synthetase 1, or CPS1 for short. Then, carbamoyl phosphate and ornithine combine to form citrulline via the enzyme ornithine transcarbamylase, or OTC. Citrulline then comes out of the mitochondria, goes through the urea cycle, which ultimately ends up as urea. Urea is then transported through the bloodstream from the liver to the kidneys, where it's ultimately filtered out and excreted in the urine. So if you follow my mouse here, this is kind of what I was explaining here. This glutamate here is broken down via GDH, or glutamate dehydrogenase, and the ammonia comes out. This ammonia is then combined with bicarbonate and 2 ATP to form carbamoyl phosphate via this enzyme CPS1. And then it's combined with ornithine to form citrulline, and then it goes through the urea cycle here, and then it's ultimately forms urea here, goes down, and it's excreted in the urine. Another way ammonia levels are maintained is through glutamine synthetase. This enzyme catalyzes the reaction where glutamate and free ammonia are combined to form glutamine. Glutamine, which contains 2 nitrogen atoms, is a major nitrogen carrier in the body compared to glutamate, which only has 1 nitrogen atom. This makes glutamine crucial for managing nitrogen levels. In the context of ammonia homeostasis, glutamine synthetase plays a vital role by converting excess ammonia and glutamate into glutamine. This process helps reduce ammonia toxicity in the bloodstream and can occur in nearly all tissues throughout the body. After this reaction occurs, glutamine would be transferred via the bloodstream to the liver, where glutaminase will break down glutamine and water into glutamate and ammonia. Then the whole process using GDH occurs, where ammonia and ammonium will be converted into urea and ultimately excreted. So I'm going to come back to that, but before I do that, I'm going to be talking about how nitrogen is connected to gluconeogenesis in the fasted state. So in the fasted state, nitrogen transport and gluconeogenesis are interconnected processes that help maintain blood glucose levels and manage nitrogen balance. During fasting, the body breaks down muscle protein to release amino acids into the bloodstream. These amino acids are transported to the liver for various metabolic processes. In the liver, amino acids from the bloodstream are used as substrates for gluconeogenesis, the process of synthesizing glucose from non-carbohydrate sources. This is particularly important during fasting when glycogen stores are depleted. The liver then plays a dual role during fasting by using amino acids for gluconeogenesis and managing the nitrogen released from these amino acids. The ammonia produced from the amino acid catabolism is then converted to urea via the urea cycle in the liver. The amino acids that become deaminated get converted to pyruvate, or one of the chemical compounds in the citric acid cycle, which ultimately become oxaloacetate, which then goes through gluconeogenesis to form glucose. So oxaloacetate is one of the beginning chemical compounds in gluconeogenesis, and either pyruvate gets converted into oxaloacetate, or one of the chemical compounds in the citric acid cycle ultimately will become oxaloacetate, and that's how gluconeogenesis takes place in the liver. And then, but since these amine groups are coming from amino acids, then ammonia builds up as well in the liver, which then goes through this process here to be excreted as urea in the urine. So this patient, the enzyme that this patient is deficient in is ornithine transcarbamylase. A deficiency in this enzyme will lead to hyperammonemia, elevated uric acid in the urine, and low citrulline, which is all concurrent with how this newborn has presented. The least affected enzyme in the urea cycle would likely be arginase. Arginase can still function rather smoothly compared to the other enzymes in the urea cycle. Because this patient is deficient in OTC, citrulline is very low. This would directly affect arginosuccinate synthetase because of the two compounds it catalyzes. One of them is very deficient. Because of the two components it catalyzes, one of them is very deficient. That would be citrulline compared to aspartate. The same downstream effect occurs for arginosuccinate lyase. However, for the last enzyme, arginase, arginine comes from other pathways and dietary sources. Talking about this enzyme here. Arginase would likely still have arginine readily available to convert to ornithine and urea. However, at the same time, arginine does become an essential amino acid. This is because normally the urea cycle and other metabolic pathways can produce enough arginine so that it is an essential. However, now that the urea cycle isn't producing arginine, other metabolic pathways can't produce enough arginine, making it essential, which just means that you have to get it through the diet. So to kind of explain what's happening in this patient, I kind of went through this already. This glutamine is going to be broken down by glutaminase, hang on. It's glutaminase, yeah. It's getting broken down by glutaminase to glutamate, which both of these cases, they release an ammonia. And then this ammonia combines with bicarbonate and 2-ATP to form carbamoyl phosphate. This is kind of where one of the problems stems from. But normally, carbamoyl phosphate will combine with ornithine to form citrulline. However, this enzyme right here, OTC, or ornithine transcarbamylase, is the problem in this patient. So, without function in this enzyme, citrulline will be very low because carbamoyl phosphate and ornithine can't be combined because this enzyme isn't working. So this citrulline will be very low. And then following everything around in this cycle, everything else would be low, very, very low as well. And then talking about how the least affected enzyme in this cycle is arginase, because arginine can be it comes from other metabolic pathways as well, and it can also come from the diet. However, the urea cycle is still one of the major contributors of arginine in the body, so that's why it becomes an essential amino acid now that this OTC enzyme isn't functioning properly. And then the other thing is why, so I discussed how this low citrulline comes to be, and the other thing, this uric acid becomes very elevated because carbamoyl phosphate, this whole process beforehand is still functioning properly to remove the ammonia from the blood, but since this carbamoyl phosphate can't be converted into citrulline, the overflow of it goes through the pyrimidine synthesis pathway, which then ultimately forms excess levels of uric acid, which is why you see the elevated levels of uric acid in the urine. And then the hyperammonemia, it's just this whole enzyme backs everything up, and that's why the ammonia gets left out in the blood, because there's so many and there's not enough enzymes to take all the ammonia out of the blood still. The next case I'm going to be discussing is case 4. Here are the facts about case 4. So it's a 58-year-old woman who presents to the ER with fatigue and weakness. The additional symptoms include diarrhea, loss of balance, numbness, and tingling in the lower extremities. Upon testing, she couldn't feel anything in the lower extremities either. So physical exam proves that she is tachycardic, has pale conjunctivas, and glossitis is present. The possible diagnoses for this patient is either it's an iron deficiency, a vitamin B6 deficiency, or a vitamin B12 deficiency. And just to get straight into it, based on the findings with this patient, she likely has a vitamin B12 or cobalamin deficiency. Vitamin B12 is a cofactor on enzymes that help with the synthesis of DNA, fatty acids, and myelin. The lack of absorption or the lack of vitamin B12 in the diet are the two primary causes of a vitamin B12 deficiency. There are other cases that are less common. For example, toxin exposure or an autoimmune condition. Ultimately, both still block the proper absorption of vitamin B12. Vitamin B12 is obtained through the diet. As I mentioned earlier, people can lack vitamin B12 in the diet, but it only occurs in people that are on a very strict vegan diet for over 3 years. This is because vitamin B12 is found in red meat, dairy, and eggs. So if you're on a vegan diet, you're not consuming these dietary things, which then leads to a vitamin B12 deficiency. Other than this, problem stems from the lack of absorption. In healthy people, vitamin B12 binds to a protein called R factor in the stomach and is released into the small intestine. In the small intestine, B12 binds to intrinsic factor so that it can't be digested. Then cells in the ileum absorb this complex via penocytosis, where it is separated again. Vitamin B12 then diffuses through the membrane into the bloodstream, where it binds to transcobalamin 2. It is then carried to the liver, where it is stored until needed. However, in unhealthy individuals, this process is failing somewhere, which results in hematologic and neurologic symptoms. In this case, this is exactly what we see. Vitamin B12 is a cofactor for the enzyme methionine synthase. This converts homocytosine to methionine, and in the process takes 5-methyl tetrahydrofolate and converts it to tetrahydrofolate. Deficiency of either folic acid or cobalamin, vitamin B12, can result in megaloblastic anemia due to inhibition of DNA synthesis, caused by decreased availability of purines and pyrimidines, thymidine monophosphate, as according to a source. Megaloblastic anemia, or macrocytic anemia, is what gives this patient fatigue and weakness. Furthermore, vitamin B12 is a cofactor for methylmalonyl-CoA mutase, an enzyme responsible for converting methylmalonyl-CoA or succinyl-CoA, or converting methylmalonyl-CoA to succinyl-CoA. Without B12, methylmalonic acid MMA accumulates. With elevated levels of MMA and homocytosine, it is hypothesized this contributes to myelin damage, leading to peripheral neuropathy. This was another sign in the patient. Other presenting complaints may include peripheral neuropathy, glossitis, diarrhea, headaches, and neuropsychiatric disturbances. All of these signs are present in this patient, only furthering my confidence in a vitamin B12 deficiency. In patients with suspected B12 deficiency, initial lab tests should include a complete blood count, CBC, with a peripheral smear and serum B12 and folate levels. This patient can be ruled out from an iron deficiency because of the peripheral neuropathy the patient was experiencing. An iron deficiency doesn't present with peripheral neuropathy. Additionally, the peripheral smear test that was ordered would show microcytic anemia if it was an iron deficiency, opposed to macrocytic anemia, which is concurrent with a vitamin B12 deficiency. The vitamin B6 deficiency can also be ruled out from the peripheral smear test because the peripheral smear test that was ordered, both an iron deficiency and a vitamin B6 deficiency present with microcytic anemia, which can rule out both of these deficiencies. However, without this knowledge, dermatitis is another common symptom with vitamin B6 deficiency. This patient had no abnormalities with the skin, yet again furthering my confidence in a vitamin B12 deficiency. Thank you for listening.